Hello everyone. Today we are continuing with the lectures on antigen antibody reactions. If you want to know more about the characteristics of antigen antibody reactions, you can go to my previous video. Here I will be discussing on types of antigen antibody reactions, in which one of the which we are covering is precipitation reaction. It is important. It is when a soluble antigen, remember soluble word is very important, when a soluble antigen reacts with a specific antibody. And this happens at an optimum temperature and pH and in presence of electrolyte. So all this can be done in vitro system. So precipitation reaction is studied in vitro test system. Why are we doing all this? To detect antibody or antigen. So one of them will be unknown and other will be known. So from known, we will detect the unknown one. So this is performed in the in vitro, where the pH and everything is maintained. And the thing is, in this soluble antigens are the ones which can be used or detected. Then only antibodies, antigen antibody reaction will form a precipitate. And this is done for immunodiagnosis of different kind of infections and the diseases caused by the infections. So antibodies which are used in it are also called as precipitants. So precipitants in this case are the ones which are used to immunoprecipitate. So you should remember that these antibodies are precipitants. And by this precipitation reaction, we can study quantitative as well as qualitative detection of antigens and antibodies, and in the in vitro test system itself. What is the mechanism? Mechanism behind precipitation as well as agglutination. Both have same mechanism, and that is given by Marek and called lattice phenomena. And a precipitate is formed when quantities of antigen and antibodies are equal. Then they will find a lattice-like structure. As you can see, this is the Y-shaped antibody combining with antigen, while antigen has antigen is multivalent and it has many epitopes, because epitopes and paratopes combine together. So many epitopes will react with antibody paratope, and there will be a bond form. And when then this all will form a lattice-like structure, then it will settle as precipitate. This happens in agglutination also. Only difference between agglutination and precipitation reaction is that in case of precipitation, soluble antigens are there. What are the different types of precipitation reaction? Precipitation reaction precipitate can be formed in solution. This precipitate can be seen in agar, and precipitate can be seen in agar using electric field. So precipitation reactions can be seen in solution, agar, as well as agar in electric field. Precipitation reaction in solution can be studied under 3 heads: ring test, slide test, tube test. Talking about ring test, in these tubes or capillary tubes containing specimen, that is usually serum. And serum, serum is having antibodies, and the corresponding antigens has to be detected. And these antigens are poured on this solution containing antibody. What will happen? And a ring will be formed, and which will be seen as a precipitate. If it's a positive reaction, that means a corresponding antigen and antibody is there. And if the test system is showing a positive reaction, what you are going to see? You will see a ring there. This is applied in the lens field technique for grouping of Streptococcus species, as well as detection of anthrax, and this is called by Ascoli's test. Let's see it in the diagram. Here is the serum containing antibody, and here when antigen is poured over it, this antigen is soluble in the solution and electrolytes are used, as I have told you. Optimum pH is also maintained here. So what will happen? This antibodies and antigen will react together and a kind of lattice will be formed. This lattice can be seen in form of a precipitate. Here you can see a precipitate. This is a positive reaction where you can see precipitate. So this is in the capillary tube, this is in the test tube, and you can see the precipitate. So antigen antibody reaction can be, and this reaction is observable, and this helps in detection in the of streptococcus and doing the lens field technique. What is lens field technique? This we will be studying in streptococcus when we will be talking about streptococcus species. This was the one which we have studied. Ring test was in a tube or capillary tube. Another type of precipitate can be seen on the slide, and this is slide test. This is known by the name of slide test. This is performed on the slides having cavity, a depth slides having a cavity in it. And what happens in this specimen, that is serum, is kept in the cavity of the slide, and antigenic solution is mixed with it by shaking. Here you can see their slides. It's this antigenic solution is mixed, and then we shake the slides. And if the reaction is positive, what will happen? You will see the floccules. These floccules will be formed. So here precipitate is seen in the form of floccules. And this is a very commonly used test. It is called VDRL also. This is Vineral Drug Research Laboratory test. And in this, what happens? Syphilis antibodies can be detected by it. And this is used for detection of syphilis antibodies. This test, if floccules are seen in tube, then it is called tube test. And this when it was done in tube, it was called Kahn's test. So when it is done in tube, it is called Kahn's test. See, similar type of floccules which we have seen in the slide can be seen in the tube itself. Another type of precipitation test is precipitation which is done in agar. So you know, agar is a solid which is obtained, it's a solid kind of media, and this agar is obtained in form of wheat which can be solidified. And how all this is done, we will be talking this in the future classes also, how agar can be used as a medium, solid medium. Here we are using agar, and there is precipitation in agar can be studied in different heads. That is single diffusion in a single dimension. That is called Oudin technique, because it was given by Oudin, so it is called Oudin technique. Single diffusion in single dimension. Another is single diffusion in double dimension. This is also called radial immunodiffusion, and this is done by Marcini Technique. This was given by Marcini, so it is called Marcini Technique also. Just students who are sitting for different kind of exams should remember the different names. It can be called by different names. And double diffusion in a single dimension is also called Oakley-Fulthrope technique, while double diffusion in a double dimension is called Audin technique. All this we are studying in detail. First one is single diffusion in a single dimension, which is called Oudin technique. What happens in this? Known antibody is mixed in agarose gel at 60 degrees Celsius. Known antibody. So what is one will be always known, another we have to, another one which is unknown has to be detected. So unknown antigens are poured over it. When this is single diffusion, because one is just immobilized or fixed inside a gel, and one is going to move. So antigen is going to move, so it is single diffusion, because one of the antigen or antibody, one is moving, other is immobilized inside the gel. So whenever, in this case, when we are talking about single diffusion Oudin technique, then it is known antibody mixed in agarose gel. Unknown antigens are antigens are poured, and if they are corresponding antigen is present with this antibody, corresponding antigen is present, you will find a zone. You will find a precipitate in the zone of equivalence. What is zone of equivalence? What is prozone phenomena? I have already told in my previous lecture when I was talking about antigen antibody reactions and the characters. So here what you will see a precipitate band. You can see this band only when unknown antigen is corresponding to this known antibody, then only this precipitation band will be seen. This precipitation band will be observed. So in Oudin technique, immobilized in the gel, antibodies was immobilized in the gel, and antigen was moving. Since antigen was moving, that was single diffusion, only one was diffusing, and it is single dimension, one dimension, because it is moving in just a single direction in a tube. So you can see this diagram is important, and here you can see the different tubes are there. Like if this is a negative test, there will be no precipitate, while in positive test you can see the precipitation, and a test will be positive only when the corresponding antigen antibody are going to react. Epitope and paratope are going to bound. So to detect an antigen, this is a very important test. Another one is single diffusion in double dimension. So this is performed on a slide, on a plate containing agar. So wells are made. Well, why I was calling it slide? Because in this, a wells are made. That is a square plate can also be used, which is plated with agar. In agar, we form wells. That means we remove agar in between. So these are called wells. And when in these wells, what happened? Antibody is there in the agar, as I have told in the previous case, and antigen is filled in this agar. When this antigen is filled in this wells, antigen is filled in wells. When antigens is filled in wells, if corresponding antigen and antibody are going to bound together, you will see different kinds of precipitates. There you can see. And with the measurement of the diameter of precipitate, you can estimate the immunoglobulins. Estimation of immunoglobulins can be done. Even detection of transferrin in serum, a protein. Detection of protein is can be done in this, and detection of influenza infection. These are the applications of these tests. Single diffusion in double dimension. Why we are calling it double dimension? Because it can move in 2 dimensions, length and breadth. Here you can see that is why a circle is formed. That is why it's a double dimension, because it can move in length and breadth both sides. While when we were doing it in this tube, only one direction length was covered. Another is double diffusion in a single dimension. This is also called Oakley-Fulthrope technique, because it was given by them, by Oakley-Fulthrope. And what happens in this case? In this case, a plain agar is made, plain agar. Here you can see. A plain agar column is given in between, and while antibody can also move and antigen can also move. Both can move. And neither antigen nor antibody is fixed in the agar. Both can move. Since both can move, that is why it is double diffusion. And antigen and antibody, as I have already explained when I was talking about antigen antibody reaction in previous video, that they have affinity and avidity towards each other. So they will move towards each other, and once they will move towards each other in the zone by following the lattice phenomena and in the zone of equivalence where both will be equal, they will precipitate. And when they will precipitate, this will happen in plain agar, because both are moving towards each other. You can see here, this will move towards each other, and then a precipitate will be seen in the plain agar. So this is another example of precipitation reaction. Another is the different types of precipitation reaction we are reading. So another is double diffusion in a double dimension, which is Oudin technique. What happens in this? How this is happening? This is a gel poured on a Petri plate. In a Petri plate, we pour a gel. Then a well is made in the center. We will be discussing that in the diagram also. Just first, I am reading this slide. I am putting all the notes also, because I want students, even if they are going for exams where they have to answer descriptive questions, so they can write directly, they can learn and write. So I keep my notes always there for all those people who will be writing descriptive answers too. So these I have made it that way. So one gel poured on a Petri plate, while one well is made at the center. Around central well, multiple wells are made. There is a well in the center, and around central well, multiple wells are made. Central well is filled with antibody solution, while other wells, which we are multiple wells, we have filled those multiple wells with antigen solutions. Now, why this is done? This is done for detection of antibodies in the serodiagnosis of smallpox. Elix gel precipitation test, important. Elix gel precipitation test for testing the toxigenicity of Corynebacterium infection. So this Corynebacterium infection toxigenicity test can be tested by Elix gel precipitation test. And what type of Elix gel precipitation test it is? It is double diffusion in double dimension. And it is also done for detection of fungal antigens. Let's see the diagram of it. This happens this way. Like this is a central well. Suppose this is a central well, and this we are showing in 3 different diagrams, but this can be done in a one plate itself to make it understand better. We are these 3 diagrams are there. Otherwise, this antibody can be in the center, while these 2 wells can be here, 2 here, and 2 here. So this whole experiment can be seen on the one Petri plate itself. So what happens in the center, we are filling antibody, and this is all over, there is agar. If these will move towards each other, and when they will move towards each other, if it is a reaction of identity, that means epitopes of 2 antigens are similar, they will fuse. These 2 lines will fuse because both were having same epitopes. So antigen was also running towards them. This was running toward, it is double diffusion in double dimensions, so 2 dimensions. So what will happen? These will show, if epitopes were similar, they will fuse at a point. If epitopes of antigen were not similar, they will just run around. They will not fuse, cross each other. And if epitopes of antigen are partially identical, this will fuse. So to study different types of antigens having common epitopes, this precipitation reaction is performed. Coming to immunoelectrophoresis. What is this immunoelectrophoresis? It is immunodiffusion combined with electrophoresis. Electrophoresis is separation on the basis of charge, and diffusion is also there. This is also done in agar, performed on agar gel, and movement is influenced by size or charge. That means in case of electrophoresis, they will move, and they will move according to the size. If the size is heavy, that will slow diffusion will be there. And also charge is also very important, because negative positive charges will move towards each other. So what happens in this? In this, we create different wells here in the agar gel plate, and in these wells, antigens are filled. Different antigens are filled, and another trough is made. There is another trough. In this trough, antibody is filled. This is a trough in the agar gel where antibody is filled. So they will move towards each other, and lines of precipitates will be formed. Those which are nearer to this will be excess in antibody. That is why line of precipitate occur here itself. That means this is more excess of antibody is there. So antigen ran, and a precipitation can be seen. Different lines of precipitate on the basis of the movement of charge and size of antigen, different paratopes, epitopes combined, and you can see different lines of precipitation. So this is called immunoelectrophoresis. This is immunoelectrophoresis, diffusion, simple immunodiffusion as well as electrophoresis combined together. Now, a step further is there, that is counter current electrophoresis. So when I introduced you first slide, it was the types of precipitation reaction, and in types of precipitation reaction, I said one in solution, another in jar, another in agar, and another one was using electricity in agar. So here we are using electricity combined with electricity is combined, and agar gel electrophoresis is there. Immunodiffusion, electrophoresis combined with electricity. I hope I am not too fast. I would love if those who are following my channel and those who are visiting my channel can just comment and let me know how I'm presenting and how I can improve myself. I will be very happy if you can comment on my and make me improve my presentations. Am I too fast or not? Please let me know. So coming back to countercurrent electrophoresis, that is Agarose gel slide is there, and it has 2 wells. Now here I have given the diagram itself because slide was having space, so I can put my diagrams there. This is a well in which antigen and another well in which antibody is poured. This is all over. Agar is there. We have made 2 wells, 2 cavity-like structure. What is well? It's just a cavity. So here we are pouring in one, antigen is there, and another antibody is there. And we are putting electricity. There is a battery also here. Battery is connected. Battery is not there in the diagram, but remember, a battery is connected over here. So when electricity is passed through this, they will move towards each other if they are having affinity towards each other, avidity towards each other. That means if the corresponding antigen is there in the corresponding antibody, then they will move towards each other. And once they will move towards each other, a band will be formed. There, here is the band, and this is faster than immunoelectrophoresis because we have combined electricity, so the charges will run fast because of the electric current, and they will move towards each other, and you can see the band. Applications are detection of antigen antibody within serum, CSF, cerebrospinal fluid, and other fluids. And it's done for the diagnosis of infection caused by bacteria, parasite, virus, fungus, etc. All this we are doing to different type of precipitation reactions are just to study different kinds of antigen, just to know infectious antigen or antibodies which are produced against that antigen in the body system. So these tests are in vitro tests for the detection of antigen or antibody. So these are serological immunodiagnostic tests. Precipitation in agar in an electric field. One more is counter current electrophoresis and rocket electrophoresis. So counter current was there. The slide has misplaced, so this should have been placed before counter current. So after this slide has come in between. So before counter current, we have to know what are the two different precipitation tests which are performed using electricity. One is counter current, another is rocket electrophoresis. This happens in the Agarose gel, as I have told that this is the gel combined, agar gel combined with electricity. So antibodies are mixed and at such pH that they won't move or diffuse. So again, antibodies are immobilized. Antigens are filled in different wells. In below, antigens are immobilized in the agarose gel, while antigens are placed in different wells, and wells are made in the gel. Only antigens diffuse under the influence of electric field to form precipitate. And bands, in this case, bands appear in form of shapes of cones or rockets. So that is called rocket electrophoresis. This is done for estimation of number of immunoglobulins. If you want to estimate what is the quantity, because it is quantitative analysis of different immunoglobulins, and also for anti-streptolysin O proteins. There are different applications also, but I have noted here only common ones. So this is the thing. If you want to see the diagram, you can see this antibody was immobilized on this agar plate while antigens were filled in different wells, and this is the rocket or cone-shaped structure which is obtained according to the concentration and availability of antibodies. I hope I was able to make this topic clear. Thank you very much.